So in this video, we're simply going to talk about nucleic acids, and we're basically saying these are biomolecules, these are macromolecules, and these are basically molecules that form up most of the genetic information that we need or that we have as species. So we have two types of nucleic acids that we're going to be interested in. So the types of nucleic acids that we are going to be looking at, we simply have DNA, and we also have RNA. So DNA is simply deoxyribonucleic acid, and we also have RNA, which is ribonucleic acid. So the difference between the two is that DNA is simply the blueprint of the information that is needed to be transferred from a parent to an offspring. Yeah, so this is like the blueprint. Then what RNA is just like an interpreter or a reader. So we say this is an interpreter of DNA. That's basically what we are saying. Yeah, so at the core of it all, whenever we are discussing nucleic acids or any type of biomolecule that we're interested in, we always start by looking at the monomer, or the smallest unit that makes up the molecule that you're interested in. So in this particular case, we're going to start talking about the structure of nucleic acids, and by so doing, we need to study the monomers that make up nucleic acids, which are called nucleotides. So we're saying we have small units that make up these nucleic acids, and those are called the monomers of nucleic acids and simply called nucleotides. So a bigger chain or a connection of these nucleotides will produce nucleic acids. So we're simply saying nucleic acids are simply polynucleotides, because they are going to be polymers formed from small units called nucleotides. That's basically what we're saying. Now, if we're to look at the structures, if we focus on looking at the structure for nucleotides, we're saying, let's say, structure of nucleotides. So hope you got the difference between the two types of nucleic acids that we have. We just said we have DNA, and we also have RNA, and we're basically saying DNA is simply the genetic blueprint of the building cells that you have, or any type of information that is needed for the production of something. In this case, we are going to specifically look at the production of proteins. Then RNA simply reads or interprets the information that DNA has to synthesize these proteins that we're interested in. That's the main difference between the two. One is the information, the other one is the reader of that information. Now, to formulate these nucleotides, or rather the structure of a nucleotide, which are simply going to make up both the DNA and the RNA. So every nucleotide is simply going to have a sugar. So we are going to have a sugar. This is mostly a pentose sugar, a 5-carbon ringed sugar. Then we also have a phosphate group. So every nucleotide will have a phosphate group. Then last but not the least, we are going to have a nitrogenous base. So this is a must. These are the three components of a nucleotide. These are a must. So it's called a nucleotide if it contains these three things that you have here. We call it a nucleotide. Then without the phosphate group, it's simply going to be called a nucleoside. So in a case, so we're just saying, take note, in a case where you just have a sugar, so if you add a sugar and a base, a nitrogenous base, you're simply going to get a nucleoside. That's the main difference between nucleotide and nucleoside. That's the difference. The difference is here in the inclusion or exclusion of the phosphate group. Now, for this sugar that we are discussing, so let's talk about how the sugar looks like. We'll talk about how the phosphate and the nitrogenous bases look like. So we're going to have, for the sugar in particular, it's supposed to be a 5-ringed structure. So for the sugar, we're simply saying it's supposed to be a 5-ringed a carbon, a 5-ringed carbon sugar. This is what you are supposed to have. This is what we expect. Of course, you have other structures. This is what you're having. So this is simply carbon number 1, number 2, number 3, number 4, number 5. This is what we mean. And we're simply saying it's a pentose. So the difference between DNA and RNA will be seen on the 4th carbon that you have. In terms of deoxyribose, which is DNA, will have a hydrogen in place of the hydroxy, whilst a ribose sugar or a sugar found in RNA will have OH, which is just this guy will contain that, this guy will contain that. This is what we mean by deox, meaning it has been deoxygenated. Yeah, so if I may give us the structures to see them clearly. So these are the structures that we have. This is still the sugar molecule. This is still a sugar. We're saying the sugar in DNA simply looks like this. The sugar in RNA is simply going to look like this. The difference is spotted here. If you notice, here there's just a hydrogen and there's a hydrogen. Whenever there is nothing indicated, just know to say there's a hydrogen. So you see the difference. This is deoxygenated, meaning it's lacking an oxygen atom. The other side is having an oxygen. And also, point of correction, the carbons are counted in the clockwise direction from here going this side. This is carbon number 1, you have carbon number 2, 3, 4, 5. This is basically what you have. So this is how a sugar structure is going to look like. Okay, so we're saying in the sugars, I emphasize the difference lies on the 2nd carbon, and this is only on the number of oxygen atoms that you have in the deoxyribose sugar and in the ribose sugar. The difference lies here on this particular position. That's basically what we are saying. So I've talked about the first component of a nucleotide, which is just a sugar. Then we're supposed to talk about nitrogenous bases. So how do the nitrogenous bases look like? So we're saying nitrogenous bases. So these are 5 in total, the bases that we're going to look at, and they are demarcated into 2 types. So we have 2 types. So then the nitrogenous bases have characters of bases because of the nitrogen they contain. So they are called bases because their pH is going to be between 7 and 14. They behave like bases because of the presence of nitrogen. That's what we're basically saying. They are demarcated. We have 2 types. These are in 2 types. We have what you call purines, and we have what you call pyrimidines. So these guys here. So we have this type, and in each category we have the difference between the 2 types of nitrogenous bases that we have is in the number of structures or the rings that we have. So these are double ringed. These are double ringed. So under purines we simply have adenine and guanine. These are the 2 types of purines that we have. So adenine is going to be represented by letter A, capital letter A. Guanine is simply going to be represented by letter G. Then under pyrimidines, we simply have cytosine represented by letter C. Then we have uracil represented by letter U. Then we have thymine, which is represented by letter T. So these are the 5 nitrogenous bases that we are simply going to be using. And thymine is found in DNA only. This guy is only found in DNA. Then you have uracil, which is found in RNA only. That's the main difference between the 2 structures of DNA and RNA. So we're saying in place of uracil, DNA will have thymine. Then in place of thymine, RNA will have uracil. That's basically what we're saying. Yeah, so in case this is confusing, we're saying the structures. Why do we call these guys to be double ringed? Because of how they look like. And if you look at the following structures, it will help us understand. So we're saying we have these 5 bases, right? These are the 5 bases that we have. we have adenine, guanine, thymine, uracil, and cytosine. The differences are just in the number of rings that we have, and we are saying these are called purines because they have two rings. You have ring number 1, ring number 2, ring number 1, ring number 2. So these are the purines. Then these are pyrimidines this side, and you can see the structures. Of course, you don't have to master the structures. We just have to know how we are going to be making them or joining them to create a nucleotide. So this is what is going to be happening. And before we even proceed, let me indicate, or rather put a note here to say this is how a phosphate group looks like, because we are from talking about a sugar and nitrogenous bases. Now we have to talk about a phosphate, because we said every nucleotide, a mononucleotide, which is just a nucleotide containing a single one of these components that are needed. So we're saying combine a phosphate and a sugar, then plus a base. What you are going to form is a single unit of a nucleotide, which is called mononucleotide, because it's just one. It's composed of these three things all the time, and we're saying they can always bond. This is the bonding you're going to see. You're going to have a condensation reaction here between the phosphate group and the sugar. This is the pentose sugar, and also a condensation reaction between the nitrogenous base and the sugar molecule, hence giving off two water molecules. That's basically what we are having. So condensation reaction is a reaction that happens to connect these three units to produce a nucleotide. That's basically what we have. And we are simply saying it's always going to be, we can show it diagrammatically written like this. This is basically a structural unit of a what? Of a nucleotide being formed with two water molecules being given off. Yeah, so this is basically what you have. Okay, so we're saying these are simply the single ringed structures. This is what you can see them clearly now, and we have the double ringed structures. So we emphasize to say whenever these guys are bonding, of course there are condensation reactions between the two parts, and this bond that is formed between the phosphate group and the pentose sugar is called an ester bond, this part here. The bond between the nitrogenous base and the pentose sugar is simply going to be a glycosidic bond. Then a combination of two bonds, if you are connecting, this is a nucleotide, maybe let me indicate it here. This is a nucleotide because you are having a base, a phosphate, and a sugar. Then you are trying to link it with another nucleotide structure here. So there are two. You link them between this this particular phosphate group is linked to the sugar that you have on the nucleotide above. This creates two ester bonds. No wonder we call this part to be a phosphodiester bond. This part here, it's called phosphodiester bond because it's formed of two ester bonds. That's basically where the name comes from. Yeah, so we're saying in a normal case scenario, we expect a sugar and a phosphate to react. So this part is reacting, giving off water. The other part is also reacting, giving off water. And this is what we are going to form. And the bond that is formed in between the base and the sugar is a glycosidic bond. And the bond between the phosphate and the sugar is simply going to be an ester bond. This is basically a single monomer unit of a nucleotide, which is just a mononucleotide with two water molecules given off. And we can proceed to say, we can look at them clearly. This is what you have. And if we repeat this process, we are simply saying here you can either have adenine, guanine, cytosine, or uracil, or thymine, depending on what you're looking at, whether, because this is in RNA. Why? Because we see there's an OH here because this is a ribose sugar. So we'll basically have uracil, right? That's what can happen. Yeah, if it's DNA, there wouldn't be an oxygen here, meaning here there will be thymine instead of uracil. Yeah, so that's basically what we are saying. So the three parts of the mononucleotide are joined by two condensation reactions, one between the phosphate and the sugar, one between the sugar and the nitrogenous base. So that's what we must know. And if we give an illustration again to emphasize on the difference, if it's under RNA, what is going to be here? There'll be OH. If it's under DNA, what's going to be here? There's going to be just H, because this is deprived of one particular oxygen. Now look at the numbering on the bases. So the numbering on the bases, on the nitrogenous base, goes in the clockwise direction. Same applies here, it goes in the clockwise direction. So this is basically what we are having. So meaning even the other molecule, so if you have two of these, this structure and something that looks exactly similar to it, we're simply going to connect it. We're simply going to say this is where we're going to be saying the 3' and the 5', the bonding we're going to be discussing is going to be coming from the carbons, the positions of the carbons that we're interested in. Yeah, so when we say 3' to 5', meaning we're going in this direction, we're going up from 3' to 5'. Yeah, that's if we're coming back like this, meaning this structure has been flipped the other way around, meaning the phosphate is down, the sugar is facing down, and the 3 is on top, while the 5' is at the bottom. So you'll see what we mean by that. Yeah, so we're simply saying in the production of these structures that we're interested in, the polynucleotides, they form very long structures, right, that are called polynucleotides, which are simply the polymers. This is simply also called a dehydration reaction or synthesis because water is being produced. Dehydration simply means removal of water. So we're saying the end of the polymer that terminates with a phosphate group is deemed to be the 5', and the other end which terminates with the sugar is deemed to be the 3'. We call it to be the 3'. The nucleic acid has a polarity conferred by its 5', phosphate group and the 3' OH group. So the polarity, or rather the charges, the partial charges that we're going to see are taken from the numbering that we've done. So we're basically saying, if I was to give us a structure here, this is what we're saying. Yeah, so the polarity of the head, let's take this to be the head, depends on this guy, which is negatively charged. The polarity of this other part, the tail, depends on the OH. That's basically what we mean. So in a case where we are to connect as many nucleotide monomers as possible, this is what you are going to have, something that looks like this. So this monomer here is formed by combining a nitrogenous base adenine with the sugar molecule and the phosphate group. Here we're using uracil, so here we're using guanine. We're just connecting these units. So let me use different colors. So it's continuing down. This is basically what you're going to have. So we're saying this is a good network of these particular nucleotides that we're having. Here they're just saying this, you should remember how a pentose sugar looks like. So if this is in RNA, meaning this is an RNA strand, this is going to be how the sugar is going to be looking like. I've just written the shortcut. This is called the skeletal structure of the sugar molecule. Then they're saying the sugar and the phosphate join here by a phosphodiester bond. So this bond that you are seeing here. Yeah. So the combination of this ester and this ester bond is what we call a phosphodiester bond. Yeah, that's basically what you have. So this is a typical example of a polynucleotide chain, a single strand of polynucleotide with ribose sugar and nitrogenous bases, and so on and so forth. This is what you have. So in a case where you are dealing with DNA, meaning the sugar, a DNA polynucleotide chain showing this here, the sugar should have been with, this should have been deoxyribose. They should have indicated deoxyribose here. Deoxyribose with only H without the O. This would have been the case. Yeah, where there is uracil, there is now thymine, right? That's basically what you'd see. Yeah, so now we're saying if we look at RNA in particular to discuss its functions, we are simply saying RNA is similar to DNA, but with two major chemical differences. Yeah, so the difference that you're going to note is the presence of a hydroxyl group that makes it to be so, the presence of a hydroxyl group which makes it to be more oxygenated than DNA. So in DNA, a hydrogen atom replaces the whole hydroxyl group. The OH minus is a game changer when it comes to the structure of the sugar molecule that you have. So this is the first, the major difference in the structures that you're able to notice. Then secondly, in RNA molecules, we use uracil instead of thymine. So now this will bring about changes in how RNA behaves, because uracil, in as much as they have the same structure, they differ on the position of the methyl group that you have, the alkyl group that is there. So if you go back, if you remember what we were looking at on the structures for thymine and uracil, so we are saying, if you recall or retract, look at what you have between thymine and uracil. Look at the position. So here we have the differences between these two is the existence of this thing here, the alkyl group, the methyl group. Here there's hydrogen in place of a methyl group. So the methyl group is certainly more reactive than the hydrogen, and they're all bonding to a double bond. So meaning we would suggest that the way thymine is going to behave, or its reaction, is going to be different with uracil, because this guy is going to certainly be more reactive than uracil if we consider this carbon number 1, the first position, where we have methyl and here we have a hydrogen. So that's a very striking chemical difference that is going to be caused because of the existence of the methyl groups. That's basically what we have. Then another thing that we can talk about in terms of the functions, we're saying RNA is not only found in one particular form, it's going to be found in different structures. So we have transfer RNA, which is called tRNA. We also have ribosomal RNA, which is RNA. We also have messenger RNA. Yeah, so it depends on the functions that you want the RNA to do or to carry out. It will take a different form. Yeah, so we're saying these are the noticeable differences, or rather that distinguishes RNA from DNA. These are the things that we are discussing here. So RNA molecules form single strands. This can fold into complex structures and shapes, or remain long, because these are just copies of the DNA. Yeah, it's just a single template of your DNA structure. RNA is produced by transcription, meaning copying, right? To transcribe simply means to copy from DNA. And these are always going to be single-stranded. The role of RNA in cells is quite varied, meaning it varies depending on what you want it to do. Like I said, there is the carrying of information. This is done by messenger RNA. It's part-time in the ribosome in the form of ribosomal RNA, and it carries amino acids, meaning it transfers things as well. So it can carry a message. It can be found in ribosomes. It can also transfer information, or rather carries nutrients, or rather amino acids, between cell components. So this is all talked about with reference to a cell, right? Whether we talk about prokaryotic or eukaryotic cells, this is basically a simple function that RNA does. So under transcription, just a quick overview of what we're going to do in transcription, because it's a topic on its own. Here we're looking at nucleic acids, and we're saying transcription is a synthesis of mRNA. We produce RNA, but the type of RNA we're producing is messenger RNA, which is basically a template of DNA. It takes place in the nucleus. You remember how you used to draw the cells in high school? You have the cytoplasm, the cell wall, the cell membrane, and the nucleus somewhere. Yeah, so in the nucleus, that's why you find mRNA. And it's catalyzed, so we're saying it forms through a catalyzed reaction by RNA polymerase. So RNA polymerase is simply the enzyme responsible for the formation of mRNA. For the transcription to happen, it's carried out by RNA polymerase. So the mRNA moves out of the nucleus to the cytoplasm into the surface of the ribosome. This is where the protein synthesis occurs. So mRNA now goes into the ribosomes. When it reaches there, it becomes tRNA. It becomes RNA, I mean. So mRNA in the nucleus is produced. It moves to the cytoplasm, until it meets, or rather goes on the surface of ribosomes, and it becomes rRNA, ribosomal RNA. That's basically what you're going to have. To carry out certain information, sometimes it carries with it amino acids, meaning it changes forms. It can now become tRNA. This is what you can have. So we're basically saying RNA is simply a component of genetic information that reads your DNA. So it's just a mirror image, a complementary strand of DNA. That's basically what we are saying. Yeah, it transcribes. So let's say we're saying we have a parent. To transfer the features that we need, the parent has DNA, right? The parent has DNA. To transfer some of the parent's features to the offspring, things like, let's say, the nose, the shape of the nose, the skin tone or something, whether it's a phenotype or a genotype change that you want to see in the offspring, what causes the information to be copied correctly, simply what does the proofreading and everything is the RNA. RNA will simply copy everything that is on the DNA and then put it in a form that is going to be read on the other side by particular enzymes and other components of the cells, until a replica of what you had in the DNA is also made on in the other species, which is the offspring in this particular case. Yeah, so this is a basic explanation of how RNA works like, but we'll have to talk about the process in which it's formed, that is on transcription and translation. They are topics, independent topics on their own. Yeah, so this is basically what I was explaining previously. So this is a quick example if we are to look at a strand for RNA. This is just a copy of some particular DNA strand. So what I mean is DNA, if we talk about DNA in DNA and the structure, then I'll give us an example on how we can quickly or easily form RNA. So DNA structure and functions, we're saying DNA molecule is made up of two polynucleotides twisted around each other. The sugars and the phosphate form the backbone, the phosphodiester bonds that you have. Then the bases point inward, so the bases are going to form they're going to form something that looks like this. This is basically what you're going to form. The bases point inward, so this is what we mean. In as much as it is twisted, there are always going to be hydrogen bonds between these particular bases. So this is a very nice illustration that we can have. If we were to just assume they are not twisted, it's not a twisted strand, it's just straight, this is what you're going to have. Yeah, so what happens in between the bases, these are hydrogen bonds that you are able to see. These will be hydrogen bonds, these will be hydrogen bonds. These guys that you are seeing there. Yeah. So now we have added to the list, aside from having a glycosidic bond and an ester or phosphodiester bonds, now we also have hydrogen bonds. Yeah, so this is a quick illustration of what can happen. Here we emphasize to say, in a DNA molecule, adenine bonds with thymine. So this is what you have. Guanine bonds with cytosine. So this is basically what you are going to have. So this is a very common thing that you are supposed to get used to if you are to form these strands. Yeah, so these strands are going to run from 5 prime, they go up to 3 prime. Then here you have 3 prime up to 5 prime, because they run in opposite directions. That's basically what we say. Yeah, so if you have a particular strand, for example, what we have here, if I want to recreate this, so we say DNA is double-stranded, right? This here is DNA. If I want to make RNA this side, I'm simply going to copy using this template here. So A will go to uracil, C will go to cytosine, C will go to guanine, T will go to this is going to be this is thymine. There's no thymine in RNA, so we have T will simply go to uracil. That's basically what you're going to have, or adenine. So we can just say uracil or adenine. This is also going to be uracil or adenine. Then this is going to be uracil. Then this is going to be G. So I'm saying this here will be the structure of RNA. DNA is double-stranded. You are seeing these guys, they are paired. But RNA is just a single strand. It's just a template to copy a certain part of the DNA that you had. This is basically what we were implying. Yeah. Yeah, so all these structures, or everything that is discussed here is just pointing out to have a structure that is double-stranded. The bases that are lined up either running from 5 prime to 3 prime, or also 3 prime to 5 prime. We basically use a word for that. We say these are going to be in the anti-parallel direction with each other. That's what we mean. And you can discuss the length between a single particular nitrogenous base to the outermost part where the phosphate groups are. This is 1 nanometer. Then the length of this particular thing, if you have one complete twist, it can be found. The length is simply going to be 3.4 nanometers. The distance between two particular bases on the single strand is simply 0.34 nanometers. These are always asked when it comes to multiple choice questions, so this is a must know. Yeah, so I just wanted to summarize this. Looking at the picture more closely, you're able to see that these are the nitrogenous bases here, right? These are the bases, thymine and adenine. Then you have cytosine and guanine. These are hydrogen bonds. Then these are ester bonds forming a diester. This part is a diester bond. Then you see between, you should be able to see that between this sugar molecule and this nitrogenous base, what you have here is a glycosidic bond, right? That's basically what you're going to have. Because the sugars are deoxygenated, this is going to be a particular example for a DNA strand. They emphasize how it looks like. This is the same thing that we are discussing. Just put more emphasis on the structure, but it's basically the same. Yeah, so we're basically saying double-stranded DNA twists into a helix with two topological features, a minor groove and a major groove, meaning there's going to be a major one and a minor one. So what we mean here is that one strand was formed from the other one. So to pair it up, meaning there's always going to be the original template and the second, the new template. That's what we basically mean. Yeah, so the two strands of DNA appear as wedges or separated by a trough. These contours between the two strands compose the minor. So there's always going to be a part. If you so wish, you can look at it to be in this context here. At every part, there's going to be like this part which is outside. This is the major part. The part that is at some point in the inside part is the minor part that we are indicating or that we are mentioning. Yeah, there's always going to be one part which is going to be on top of the other. So depending on the portion of the DNA that you are focusing on, one part is going to be minor, the other one will be said to be a major one. Yeah, so the variations can really only depend on the sequence that you're going to have, because sequences are going to be different. This explains why we all have different faces, different noses, because there are so many combinations of these nitrogenous bases, which is obviously in turn going to change the information that is read by RNA, right? That's basically what we're saying. Yeah, so we're saying the DNA in animal cells is highly compressed in tight structures with the aid of DNA binding proteins called histones. So histones are simply components, or rather things that help to make DNA to be so compact. DNA is very big. If you were to stretch it, the strand is very long. But how does it stay compressed in cells? Because of histones. It's stabilized by histones and it's twisted, very complex. Like the same way you talked about when we were discussing proteins, quaternary and the tertiary structures. That's how this guy is twisted. The long strands of DNA wrap twice around the barrel-shaped histones until a structure resembling a strand of pearls is formed. These strands are then twisted and folded onto highly compressed arrangements. So that's basically what we are saying. So it looks like this. This is what you have. It's very, very, very twisted back and forth. Yeah, so that's basically what we are saying. Yeah, so between the breaches that we're talking about, we're saying the bigger portion, if you look at if there's a major difference in the depressions of these guys, so let's call this to be your You used to call, in physics, if you have waves like this, you call these troughs, right? These are the troughs that you have. So these are the crests. These are the crests. Then the down part are the troughs, right? So these parts here, if you have major depression, we call it a major groove. If you have a small depression, you call it a minor groove. That's basically what we're going to have if it's put in a ribbon form. Yeah, so these are just the same thing represented in different ways. So the double helix, so the bases are going to be complementary because they're always going to be, this strand is going to have the other strand. They are pairing up the bases, so that's what we say, it's a complementary base pairing because we just pair them up. Yeah, so A to thymine, this is going like that. This guy is they are switched here. One is going down, this one is going down. You're seeing the arrow, it's 5' to 3'. The other one is 5' to 3'. So they are anti-parallel. We emphasize that particular point. So this is basically what we mean. And we're saying the naming comes from the position, or rather the carbon positions on the sugar molecules. This is 5 on the 5th carbon to the 3rd carbon. So we're simply saying this is simply what? This is simply the arrangement, and it's going in this direction. So this is what we have. Yeah, just to summarize all this, we'll talk about, because everything is just repetition here, there are other vital components of these nucleotides. Nucleotides don't only form the two structures we are from looking at. Sometimes, or in certain conditions, we can have use of these guys in the body reactions, in the cellular reactions that we have, in terms of having vitamins, or rather having energy. So ATP simply means adenosine triphosphate, right? That's what we basically mean. Yeah, so we just have, this is a nucleoside with the 3 phosphate groups. That's what we basically mean. So if we say AMP, we simply mean adenosine monophosphate. So the naming will come from the nucleoside. Remember what we said a nucleoside is. A nucleoside is a combination of a base and a sugar. That's a nucleoside. So if we add a nucleoside with a phosphate, these are the combinations that we'll be getting. These are the names. This is where the names are coming from. Yeah, to just wrap up the whole thing, we're saying these are the major differences between DNA and RNA. We're saying DNA has bases adenine, thymine, guanine, and cytosine. RNA has adenine, uracil, guanine, and cytosine. So in DNA, A bonds with T and G bonds with C. Then in RNA, A bonds with U, G bonds with C. This is a deoxyribose sugar, a deoxygenated sugar lacking the oxygen or the hydroxyl group. This guy is very oxygenated. It has the hydroxyl group. This is the original information that you have. This is just a copy. There's only one type of DNA, right? This is basically going to have many forms. This is what you have: mRNA, rRNA, you have tRNA. So these can take 3 different forms. Then this is primarily found in the nucleus or chromosomes, and it's very large, double-stranded. The opposite happens here. It's spread throughout the cells because you can find it in the cytoplasm and also on the ribosomes, of course in the nucleus as well. And we're simply saying this guy is only single-stranded. So this is basic nucleic acids. These are the informations that you need to know, and this is the information you need to have under nucleic acid. So if you have any questions, feel free to inbox me. In case I mentioned something that is contradicting what you saw somewhere or some other book, you can just feel free to WhatsApp me so that we reach a mutual agreement on the concepts pertaining nucleic acids. Thank you so much for watching. I'll see you in the next video in transcription and translation.